Epistasis occurs when the expression of alleles at one locus alter or inhibit the expression of alleles at another locus. This phenomenon allows multiple different genotypes to display the same phenotype. Single recessive epistasis is the result of the homozygous recessive gene trait at one locus affecting the expression of a trait at the second locus. In this example, we will be exploring two heterozygous chocolate cakes that combine ingredients to produce varying offspring. Both parent cakes are represented by the genotype big A, big B, little b. In this case, the A locus determines the presence or absence of chocolate in the cake's phenotype. The dominant A allele, represented by big A, or big A, indicates chocolate is present, whereas the homozygous recessive A allele, little a, indicates the absence of chocolate. Therefore, the cake remains vanilla, regardless of the genotype at the B locus. The B locus represents a variant in the amount of chocolate present in the cake. The dominant allele, big B, or big B, little b, indicates the cake is completely chocolate, whereas the homozygous recessive allele, little b, indicates a marbled appearance. To determine the proportion of phenotypes displayed by the offspring, we begin with a dihybrid cross. A Punnett square for each allele is written separately to determine the probability for each genotype at both loci. Once each probability is determined, we can then create a branch diagram as seen below to combine the probabilities of each allele. We multiply the proportion of each A allele by the proportion of each B allele to determine the resulting phenotype ratios. 9 out of 16 genotypes result in the dominant AB phenotype. 3 out of 16 result in the dominant A and recessive B phenotype, and 4 out of 16 result in the homozygous recessive A phenotype. This 9 to 3 to 4 ratio is indicative of single recessive epistasis. Here we have a visual representation of the data displayed on our branch diagram. There are 16 slices or offspring with varying genotypes. Those with higher probabilities are seen repeated in the visual. The 4 slices highlighted in red display the homozygous recessive allele at the A locus, indicating an absence of chocolate no matter what occurs at the B locus. The 9 chocolate-covered slices indicate the dominant allele is expressed at both the A and B locus, where the presence of chocolate is determined by the big A and the dominant chocolate variant is determined by big B. The 3 marbled slices indicate the presence of chocolate at the A allele, but the homozygous recessive variant at the B allele, resulting in its marbled appearance. You can see the 9 to 3 to 4 phenotype ratio expressed in the offspring. Using the same example, a second cross is performed. Similar to the first example, the probability of each genotype at the A locus is multiplied by that at the B locus, displayed on the branch diagram below. Half of the resulting genotypes display dominance at both the A and B loci. The other half are homozygous recessive at the A allele, resulting in a 1-to-1 phenotype ratio. The half highlighted in red are homozygous recessive at the A locus and therefore do not display the trait for chocolate. Again, in this case, the variant at the B locus is irrelevant. The chocolate-covered half contains the dominant allele at both the A and B locus, resulting in its chocolate-covered phenotype. No genotypes that are homozygous recessive at the B allele are present, and therefore none of the offspring display a marbled appearance.